And full screen.
Yes, I'm doing it. I couldn't con others into doing it. And I assume that you're seeing my screen.
Yep, full screen looks good.
Good, good. Yeah, before I start into the scientific talk, since I'm in Texas, in Houston, I do want to note that as we speak, funerals are going on in Uvalde, Texas, as they did recently in Buffalo, New York. We all know why. We all know what it means. And if we're looking for a bright spot, then you probably have seen in the last day that Justin Trudeau, Prime Minister of Canada, has announced a ban on the sale of handguns. And there's a proposal in the works to ban possession of assault rifles. So it is possible, and possibly, though unlikely, the bipartisan committee now active in Congress will do something at least incremental to help us south of the border. So with that, let me turn to the science. I'm going to talk about a piece of software and an environment that we've created, the Metabolomics browser, which you see in screenshot here. First of all, acknowledgements up front. As I said, others demurred when on giving this presentation. I'd like to emphasize the work of Bradley Broom and Rehan Akbani, and also Philip Lorenzi, who is the director of our metabolomics and also proteomics mass spec cores. And then Iqbal Mahmoud, who is on the line here, who is leading our charge to shared omics with DNA, RNA, and protein to combine with what I'll be describing you today. I'd also like to emphasize our fine software engineering team headed by Chris Wakefield. But since this is a session on collaborations, even more so, the group under Shankar Subramanian, including Owen Fahey and Mano Moria, with whom we're working on this project. And of course, our principal funding from the NIH Common Fund Metabolomics Program, as well as from two foundations, Chapman and Michael and Susan Dell, who partially supported the work. Well, it starts with the workbench. This Owen has already mentioned web-based tools. Let me blow up that section. The MetaBatch omic browser that I'll be describing, and one of its components, what we call next generation clustered heat maps and their viewer. This sits at the 70,000 foot level of what the architecture looks like, with emphasis on input from Metabolomics Workbench, but also from our local data and other metabolomic databases as well. I don't think I have to describe very much in this overall diagram. There's the computational API, both for custom batch assessment and detection tools, and then for custom batch correction tools. Heavy emphasis on visualizations of several sorts that I'll be describing to you. But first, I'd like to make a couple of statements about batch effect analysis and correction. First, it's more than just applying an algorithm. It usually requires detective work for diagnosis and interpretation, usually in concert with or by domain experts, particularly to distinguish technical from biological batch effects, partly through fluent, dynamically interactive visualizations, which can be very helpful in the process. So that's going to be a theme in what I describe, particularly for discovering batch differences, biological or technical, that had not been pre-assigned. The tools that I'll be describing do that, even if one has not been prescient enough to define the batches that are most at fault. So here's what it looks like in a slightly different view from the metabolomics workbench. The data are entered into a standardized data metabolomics workbench tool, standardized data format, a metabatch analyzer. There is a batch package, which started a batch R package, which started with mbatch for the other omics, but then major customization and alteration for metabatch. And then the results are expressed in the metabatch omic browser, which I'll show you in a moment. Most recent pipeline runs downloaded all available workbench data, not quite the 2,091 data sets from 863 studies that Owen mentioned. So we're slightly behind, but not very much. It took 7 days to analyze using 15 cores on our compute server. Now, I'd like to emphasize the work of a fine scientist who I've been sharing efforts with recently. That's Gary Patty, June 1st, 2022. I really like this slide and what he had done in optimizing the algorithms for batch correction, showing a great advantage to random forest. I'm sure he knows very well that there's no one size fits all. Machine learning or AI, deep learning or statistical methodology or algorithm, it really depends on the nature of the data, distributions, et cetera, et cetera. But this is really a nice comparative study for sure. I will not be talking about algorithms today. We also explored a number of them, including those that involve deep learning. The database that I'll be showing the results from is the TEDdy database on diabetes as analyzed by Oliver's group and collaborators. And this is what the initial output of the metabatch omic browser looks like with this, relatively speaking, large data set. When I say relatively speaking large, I mean because we've also been working with single cell databases, which have gotten up to the point of having about 200,000 columns representing individual cells. So for this purpose, we're not at that level for the metabolomic data sets, even the large TEDdy one. This is a principal component analysis. And now let me show you a short tour of what the output looks like. First of all, here is the compendium of data sets, almost 2,000 of them represented here with search functions, quite obviously for location of ones of interest. As I said, we are not quite at the level that Owen mentioned, but we'll be updating. The upper left of the panel is a set of controls for choosing algorithms for representation and a variety of kinds of information, including details on the data sets. This is immediately accessible from the link in the Metabolomics workbench that I showed you earlier. And here's the central panel, which includes the PCA in this case, and then tabulations for the various batches that have been defined, and a batchometer, as Rehan Akbani has called it, which includes the DSC, a discriminant metric used to assess the intra versus inter batch statistical characteristics. Second kind of visualization is our next generation heat maps. And I think I'll have time at the end of this to show you very briefly what it looks like on the fly. Key features are extreme zooming without loss of resolution for drill down to the point at which in even a very large database, 100,000 elements on a side, one can see the labels and use them in a way and use them for link outs to dozens of other external resources. Fluent Google Map-like navigation, link outs, as I said, to many different resources, including the ones listed here for metabolomics, as well as other omic disciplines, recoloring on the fly, high resolution graphics that meet the criteria of all of the major journals, annotations with pathway data, and then the saving of all metadata needed to reproduce any state of the map. That's in addition to other kinds of functions. Overall, it's a multifaceted exploratory environment for analysis and interpretation of metabolomic, and also with, as I mentioned this morning, I have characterized in the distant past as integromic data, and others would call multi-omic. As part of it, let me introduce you to what we've called Omic Pioneer, which is the NGCHM, Next Generation Clustered Heat Map, but then tightly bound to sample-wise information such as a UMAP, a TISNI, PCA, PCA+, or MDS. And also on the other side to pathway and potentially to gene ontology and similar kinds of information. I won't go through all the names of the people who have been seminal in producing this, but suffice it to say that if you find something that's of interest to you in a UMAP, you can then project it into the corresponding genes and samples in the heat map, and then into the information in the pathway. Speaking of UMAPs, this is a UMAP of what I showed you before in PCA form. And again, you see there's a small outlier. It doesn't correspond to any of the pre-assigned batch parameters, despite what it looks like in this figure. And we are investigating what may be the cause. Also, incidentally, there is a dynamically expandable form of box plot and violin plot and other ways of looking at the data as well. So back to where I started, this is the overall diagram. One of my close friends from the NIH said once, that largely in jest, but I'll reproduce it anyway, that unless you publish something at least twice, no one is going to think you really mean it. Well, we really mean it about batch effect analysis and correction, more than just an algorithm, detective work, usually by domain experts to distinguish technical from biological effects, for fluent, dynamically interactive visualizations, which can be helpful for discovering batch differences, biological or technical. Metabatch, we feel, provides a dynamically interactive, highly visual framework for exploration and discovery in the metabolomic and integromic spaces. So with that, let me cite something that was said by a scientist who we have very much enjoyed working with very productively. He said, "We want to stay in close touch with the other omics." That was Yves Fahey in Zoom Heaven, June 1st, 2022. We're very pleased to be working with him and with Shankar and with the team here that's listed. And Moria missed this incarnation of the acknowledgements slide. So with that, since I do have some time left, And I assume everybody's on the line that I haven't been talking into space. Is that correct?
That's correct.
Hear you, John.
Okay, all right. I'm going to stop sharing the slides and use a few minutes just to introduce you to the machinery that I've been talking about. You can look for yourself at the Metabolomics browser, but let me just go back to what we've been working on for a number of years, which is the next generation cluster heat maps. And I'll try to at least indicate to you why we think it's a tool which we're using very extensively. For example, Rehan Akbani in our RPPA proteomics core, and for many of the TCGA projects and their descendants. On the left is a relatively small next generation clustered heat map. This is 3000 by 3000. There are much larger versions of it. This is down sampled. Covariate bars, which you can see in the dendrograms. And over in the very upper left-hand corner, you see a little green rectangle. Well, that represents what's in the detailed view on the right-hand side. So let me just take it to to what I think is an interesting point. And you can see that it would be of interest to an immunologist because it contains a variety of HLA molecules. And I apologize, this is the best example I have for showing the features, even though it's not on metabolites, but we do have equivalent, though much smaller, for metabolites as well. So we can do all the things you'd expect. You can select regions from the dendrogram, can zoom and navigate, zoom to the point in which one can read the labels, choose a label, and I'm right-clicking on it to show up a variety of the kinds of additional sources to call on. For example, if we're interested in 3D structure, then we can, oops, I pressed the wrong one. If we're interested in 3D structure, then what I meant to hit was Muppet. So one can do all the usual things with 3D structures and so on and so forth, a couple of dozen of them and a growing repertoire. In addition, then, one could, for example, add a pane below this one and choose to put in it the 2 or 3D scattergram. Let's do a 2D scattergram. Apply, and this would give us a UMAP or a Tisney or whatever sort of 2D figure we wanted. A variety of kinds of controls, a couple shown here for optimizing the view. And then one could select a set of cells and would set a set of cells, and it would project into this figure where those cells are and can be used for annotation of the cell types. Very useful is to be able to zoom asymmetrically, for example, to keep all of the width in view in order to scroll through the data set. Recoloring on the fly. One can put layers of these heat maps behind each other, for example, to compare RNA and protein expression, a variety of ways of saving the graphics and of saving the database in order to show it months or years later. So I'll stop there. That just gives you a feel for what I meant by dynamically interactive exploratory environment. And I'm happy to take questions if there is viewed to be time to do so.
Thank you very much, John. Yes, you've gotten us back on schedule, which I very much appreciate. We actually have about 5 or 6 minutes for questions. So John, do you have metabolomics data readily available in the tool that you can kind of walk us through how the clustering and what have you works with that?
In the cluster, well, here, what I'm showing you now is that, let me go to the NGCHM. This is again the TEDdy data set. And here is the same thing. Is that what you meant?
Mm-hmm.
Yeah, it has not been updated in this public release, and this is all public. It hasn't been updated to include the 2 and 3D UMaps and such, but otherwise it's pretty complete.
Very cool.
And all this is what you'd access if you go to the link in the Metabolomics workbench. As long as the
is there a specific software, if you know beforehand, you are expecting a particular metabolite in among the analytes? Is there a specific software which can validate your finding in the web browser?
when you say a specific metabolite to validate, when you say what kind of finding are you referring to?
I'm just referring to the identification and the structure of it, especially when you're expecting it.
That's a good question. Generally speaking, if we are going to be doing searches based on the metabolites, we would be using their RefNet names or some such, and would be using it at the level after the pre-processing of the spectrum so that we had a putative identification. This isn't a tool which is aimed at aiding in the identification process. Does that answer your question, or did I misinterpret?
No, you're right. Thank you. Yeah.
Now, obviously, it can also be used and used at the level of particular peaks if one wanted to, but that's not what we principally designed it to do.
Thank you.
Any other questions for John?
I guess I have a question. My question would be, is there anyone there who feels that this is something that they'd like to explore because it might be useful for what they're doing?
John, I think I'll speak for our group here. We would definitely like that. We actually had a question about whether or not it's compatible if you could upload a CSV file or CSV file.
The inputs, Bradley, what are the input types that can be used?
A CSV file up into the CHN builder, and then that creates a compiled version of that data, which you then load into the viewer.
I'll also say for most of the tools we're developing, we have GUI-driven versions. In terms of the heat maps, we have a simple GUI-driven version with an advanced button on it. We then have a much more complex advanced builder. Then those, like our analysts who really prefer to work in R scripts, not in GUIs, we have an R version. And then for those who really want to get into the guts of it, then the JavaScript, et cetera. And implemented in containerized form, and we have implemented on the cloud in a couple of instances as well. Bradley, do you want to say something more about the architecture or the computer science aspects of it?
I wasn't planning to.
Okay, all right. Or Rehan or anyone else who's on the line from the group?
I had seen that Amanda Shaver had turned her camera on. Amanda, did you have a question?
I don't, but thank you.
I think this is a very friendly program for visualizing metabolomics and lipidomics data. I see the question in the chat box asking CSV file. Yes, you can simply upload CSV file, but there are some format you need to do, like group wise and sample wise. So I think you can just simply upload and visualize and see dynamic features throughout the heat map. Yeah, it's very friendly program.
And let me introduce Iqbal, who I had mentioned earlier, who is leading us in the multi-omic integromic aspects of this. He's developed some lovely ways of looking somewhat differently at pathway data. And it's not yet in public release, but we're headed that direction for combining the RNA, protein, and metabolomic levels.
Well, we certainly look forward to that, Iqbal.
Thank you.
With that, I think we will switch into break mode.